global perspective on the FLORA data, and has it changed practice? We'll start with Switzerland. Is this uptake now happened, and are you using it frontline?
Yes, it's a standard treatment frontline. I think we have to be really convinced. Of course, we wait for the survival data. All survival data with TKIs are always fully conditioned by the crossing over. So we have to keep in mind, look at the data and look at the final crossing over, meaning the potential confusion which is given by receiving the drug later on. It's very important to look at all these parameters to evaluate OS, but the PFS is convincing. We have to keep in mind that we like to add, the PFS of frontline, erlotinib, gefetinib, and the PFS of second-line osimertinib, but we don't speak about the same patient. We have been giving, and I agree, osimertinib only in patients with T790M in second-line. So half of these patients, if you don't give them it frontline, will never be exposed to osimertinib. And osimertinib, by the way, in terms of establishing new standard, is extremely well tolerated. We still have some patients with difficulties in tolerating erlotinib, gefetinib, or even worse, afatinib, or don't even speak about dacomitinib. So it's really a drug which allows for a good quality of life and a very decent kind of restart of a normal life, at least for some months, right?
Yeah. Big discrepancy in toxicity with first-generation TKIs.
Absolutely.
The UK perspective, Sanjay, on this data?
Yeah, I mean, the data speak for themselves. To us, this is a great standard of care now. The challenge we have in the UK is that the discussions between the NHS reimbursement agency, NICE, and the industry partner AstraZeneca are still ongoing, and we look forward to resolution of these in the next few months. So at the moment, first-line osimertinib is not standard of care in the UK, and we're still sequencing. But I think, when we're in a position in a few months' time, hopefully when that will become available, there'll be a pretty rapid uptake for frontline osimertinib, really for the reasons that we've discussed.
And similar in France?
Similar in France, but we don't have access first line currently. It's not reimbursed, so it's ongoing. So that means we still use systematically for the patient who are T790M positive beyond first generation or second generation. We will still have the discussion about the sequence of best drug first. We just should keep in mind that we never know what will be the tumor evolution when we start a treatment. And this is a major issue. And the main difference is if we compare for the HALK population, for which you can expose all the patients to the different generation, whatever the type of mutation of resistance, which is not the case for the EGFR, for which an osimertinib, you need to prove, if you want to keep it in second line, the patient is T790M positive. And as it has been said, only 50%, and I would say in clinical practice, sometimes it's much more close to 40% than 50 or 60% T790M positive. We know that, and in all the trial and also in the FLORA trial, we have around 20 to 30% of the population that never get any second line of treatment in this population. So this is something really important. And I would say we never know the evolution of T790M positivity, and it's unpredictable, and we never know if we will able to capture this mutation, particularly if you have a brain evolution. Generally, this population have circulating tumor DNA negative. You cannot perform any tumor tissue biopsy, so you're not supposed to expose this population with osimertinib. You can have some bone evolution also. We know the challenge to make any molecular testing on this type of tumor and disease evolution. So that's why this is always what I say, particularly for this population and for the osimertinib. You cannot write the script of the patient at baseline. And so we are not playing a game. So on the paper, it might be nice to say, okay, I have 10 months, if I keep osimertinib, it will be nice. Yeah. But you never know the scenario when you start the treatment. And you have the patient in front, and you will say, okay, do we play a game? In 50%, we will have the sequence. Or in 50%, the story will stop after the first generation. And it's a short story, because after, it's mean chemotherapy. Yeah. So I don't think the fact to tell, because we can hear a lot of things to tell, okay, but after osimertinib, I have nothing, so I don't want to use it in first line. Right. But if you use osimertinib in second line, it will be the same thing. After osimertinib in second line, you have nothing beyond. And so that means currently we all have in favor of osimertinib. You have the PFS, response rate, brain efficiency, you prevent the brain evolution, and for me this is probably one of the most important things. It has been seen also with alectinib. When you see the probability to develop brain mets at 1 year, it's 8% osimertinib, it's 25% in the standard of care with gefetinib or erlotinib. So it's something major for the patient. And lastly, what we saw also, we look at the time to discontinuation of any EGFR TKI, we look at the PFS2, for which in this case you take in account 2 lines of treatment, and even for the PFS2 in the FLORA trial, you maintain the magnitude of benefit in favor of osimertinib, despite the fact that in the other arm, the patient receives sequence of the treatment. So that means whatever, you will do better to start with osimertinib than to keep osimertinib in second line of treatment in 50% of your population, and tolerability is quite nice.
Very good points. I think that you can't ensure that a patient's going to get the active drug. You have to identify the T790M, which is only in 50% of patients, and you can't ensure that that patient's actually going to get any second line drug. And I think that you've got progression that happens in the brain and systemically that unfortunately precludes a lot of patients from getting any additional therapy. So I think using your best drug first in this instance is important. Ensures that every patient gets the right drug. Some questions have come up, as you mentioned, David. People have, at least in the U.S., liked the sequencing story initially. It was a first-generation TKI. It was a neat story identifying T790M in the plasma, and then they went on to osimertinib, and of course that's being supplanted by first-line osimertinib. Now the question is, and I've asked this question before, what do we do after osimertinib? What is the common practice if we're using osimertinib frontline? What do we know anything? And Ram, maybe you can comment on this first about mechanisms of resistance to osimertinib. And does that inform treatment decisions? And do we just use chemotherapy for these patients afterwards?
So right now, the standard of care for those patients after osimertinib is platinum-based chemotherapy. But we know that that will change in the upcoming years, because we will understand more about what are the mechanisms of resistance behind osimertinib. Now, it's public knowledge now on the ESMO Congress website that next month we will have a presentation in Munich about resistance mechanisms in the blood for patients treated with osimertinib in the FLORA study. And I think those type of data will lead us to develop approaches that can overcome resistance. Just like we had erlotinib and gefetinib, and we learned about T790, and we will develop a new drug to overcome T790. We will have approaches to overcome resistance to osimertinib. So I think this is a dynamic field. We're by no means where we want to be, but we're in a much better place with osimertinib as frontline therapy than we were a few years ago.
There are good examples. There's lots of ongoing research about one of these mechanisms, and again, we'll learn at ESMO a little bit more about the prevalence, but one of the resistance mutations, the C797S mutation, is potentially giving rise to a new sensitivity to first or second generation TKIs. In the lab, there are new also developments of drug targeting this mutation, but also in some patients anecdotally. So I think we will gain this kind of portfolio basket approach at resistance, which will help us maybe to distinguish opportunity others than platinum-based chemo. But again, I think the most expensive price you have to pay if you start with an old generation is not so much this calculation of PFSs. I think we always underestimate the cost psychologically and also in terms of expenses of having brain mets. Yes. Socially, how handicapped and debilitated you can be, people need to support you, and it has really something which is the worst scenario for a patient. So having a drug being able to delay that is probably very important.
And not only delay it, but circumvent the need for radiation up front has been a huge alteration in what we used to do for these patients.